And so we come to the final topic of the quarter, the immune system. The function of the immune system cannot be overstated, especially during this unprecedented time of COVID-19 pandemic. We all hope that we, as humans and stewards of civilization as we know it, that our immune system will help us survive this present time of danger. It does not escape our notice, however, that the immune system is the most unique organ system of the body. Whereas most organ systems in the body are comprised of active workings of specific organs, either physically connected or connected by the circulatory system, the immune system comprises mainly of active cells and their products. Although they have their cellular origins from the myeloid tissues of the bones, the arena of their functions is not only a few selected organs, but anywhere where there are infections or trauma. The defense system of the body, however, is not the sole work of the immune system. We employ other parts of our body that act as barriers to invading pathogens, microorganisms that cause diseases or disorder in our body. We call this as non-specific defense systems. So in this topic, we will discuss both the non-specific and specific defense systems by which we enjoy with or without our conscious experience of it. The study of physiological defenses by which the body destroys or neutralizes foreign matter, both living or non-living, is a special field in biology and medicine called immunology. The immune system serves to protect the body against infections by microorganisms, isolate or remove non-microbial foreign substances, and destroy infected cells or cancer cells that arise in the body. The third function is also known as immune surveillance. The body's defenses is traditionally divided into two systems, the non-specific and innate immune system and the specific or adaptive immune system. The non-specific or innate defense protects us against foreign cells or substances that does not involve the tedious, time-consuming molecular mechanisms of cell-to-cell recognition pathways. The non-specific innate immune defense is like a lowly foot soldier who is always prepared, responding within minutes to protect the body from all foreign substances or cells. The system has two barricades. The first line of defense is the external membranes or body membranes, intact skin and mucosae. The second line of defense, called into action whenever the first line has been penetrated, relies on internal defenses such as antimicrobial proteins, phagocytes, and other cells to inhibit invaders' spread throughout the body. The hallmark of the second line of defense is inflammation. The specific or adaptive immune defense provides defense that depend on specific cell-to-antigen recognition mechanisms between immune cell and foreign cell or substance. The specific adaptive defense system is more like an elite fighting force equipped with high-tech weapons to attack particular foreign substances or cells. The adaptive defense response, which provides the body's third line of defense, takes considerably more time to mount than the innate defense response. The microbes that our body defenses watch out for includes bacteria, viruses, protists, fungi, and parasitic metazoans such as helminths and insects that burrow into the human skin. The first line of defense of the body includes the surface membrane barriers. This includes intact skin epidermis and mucous membranes. The intact skin epidermis forms mechanical barrier that prevents any entry of pathogens and other harmful substances into the body. This includes acid mantle of the skin. These are skin secretions like sweat and sebum that makes epidermal surface acidic and inhibit bacterial growth. They also contain various bactericidal chemicals. Keratin, also part of the skin, provides resistance against acids, alkalis, and bacterial enzymes. Intact mucous membranes form the mechanical barrier that prevents entry of pathogens. This includes mucus that traps microorganisms in respiratory and digestive tracts, the nasal hairs that filter and trap microorganisms in nasal passageways, cilia that propels debris-laden mucus away from the nasal cavity and lower respiratory passageways. Gastric juice contains concentrated hydrochloric acid and protein-digesting enzymes that destroy pathogens in the stomach. Acid mantle of the vagina inhibits growth of most bacteria and fungi in female reproductive tract. The lacrimal secretion, or tear, and saliva continuously lubricate and cleanse the eyes and oral cavity against microorganisms. Urine, normally acidic pH, inhibits bacterial growth and cleanses the lower urinary tract as it flushes from the body. The second line of defense includes the innate or nonspecific cellular and chemical defenses. This includes phagocytes that engulf and destroy pathogens that breach surface membrane barriers. The macrophages also contribute to adaptive immune responses. Natural killer cells promote apoptosis, that is, cell suicide, by directly attacking virus-infected or cancerous body cells. They recognize general abnormalities rather than specific antigens and do not form memory cells. Inflammatory response prevents injurious agents from spreading to adjacent tissues and disposes of pathogens and dead tissue cells and promotes tissue repair. Released inflammatory chemicals attract phagocytes and other immune cells to the area to increase defense. The antimicrobial proteins include interferons, alpha, beta, and gamma. This includes proteins released by virus-infected cells and certain lymphocytes that act as chemical messengers to protect uninfected tissue cells from viral takeover. They mobilize immune system cells as well. The complement proteins are a group of blood-borne proteins that, when activated, lyse microorganisms or destroy microorganisms and enhance phagocytosis by opsonization and intensify inflammatory and other immune responses. Fever is a systemic response initiated by pyrogens. These are foreign chemicals that are produced by the breakdown of foreign microorganisms. High body temperature in fever inhibits microbes from multiplying and reproducing and enhance body repair processes. All leukocytes or white blood cells, including red blood cells, develop from a progenitor cell called blood hematopoietic stem cell. The process of developing into specific blood cell is called hematopoiesis or hemopoiesis. The process is mediated by chemical signals that include erythropoietin, colony-stimulating factors, and cytokines. All of the immune cells start development in the bone marrow, but some migrate later to lymphoid tissues, like the thymus and lymph nodes, where they differentiate. RBCs, platelets, and granulocytes develop from myeloid stem cells at the bone marrow. Lymphocytes and natural killer cells, on the other hand, start development in the bone marrow from lymphoid stem cells, but later on differentiate in lymphoid tissues. In the following 7 slides, we will present the nonspecific defense cells of the immune system. We begin here with the 3 granulocytes and then followed by agranulocytes. The first granulocyte we show here is the eosinophil. This is an artist's depiction of the eosinophil. It normally comprises of 2 to 5% of all white blood cells in the blood. These granulocytes destroy multicellular parasites such as helminths by unloading their hydrolytic content of their granules on the parasite. They also help decrease inflammatory response after infection has been contained by the defense system of the body. The neutrophils are the most abundant granulocytes. They comprise 90% of all white blood cells. They phagocytize foreign microorganisms and secrete inflammatory mediators or chemical signals such as vasodilator and chemotaxin cytokines. The basophils are the least abundant in normal blood, comprising only 2% of all white blood cells. But they are very potent granulocytes. They release histamine that mediates inflammation. They destroy parasites when they degranulate. The medication antihistamine is partly to alleviate the effects of their action in tissues. Monocytes are one of the two granulocytes among the white blood cells in blood. They are distinguished by a large nucleus that is indented on one side, sometimes looking like a horseshoe. They comprise between 0.9 to 6.3% of all white blood cells. They phagocytize foreign microorganisms or cellular debris, and they often migrate into the tissues and give rise to the macrophages. Mast cells appear like basophils. They are granulated and stained with basic dyes. Their granules contain histamine and an anticoagulant, heparin. They are distinguished from basophils by their round nucleus. They migrate into the tissues and mediate inflammation and wound healing, and are involved in angiogenesis and immune tolerance. Macrophages develop from monocytes that have migrated to the body tissues. They are therefore one of the most widely distributed nonspecific defense cells. They phagocytize foreign microorganisms in cancer cells. They are one of the most important antigen-presenting cells, but can also communicate to specific immune cells by secreting cytokines, informing them about foreign invasions. Natural killer cells, like specific immune cells, develop from lymphoid hematopoietic cells. They secrete toxic chemicals into infected cells or foreign microorganisms and kill them. They are activated by macrophages and helper T cells. And lastly, but not the least, you have the dendritic cells. Dendritic cells are like macrophages. They are one of the most widely distributed defense cells of the body. They assume different names and forms in different organs of the body. They are highly phagocytic and are antigen-presenting cells. Like macrophages, they play a key role in non-specific and specific immune defense of the body. Phagocytosis is very important in non-specific immune defense. Once neutrophils, macrophages, and other leukocytes arrive at the site of infection, they begin the process of destroying invading pathogens by phagocytosis. The initial step in phagocytosis is contact between the surfaces of the phagocyte and the microorganism. Chemical factors such as opsonins in blood and tissue fluids can also bind the phagocyte to the pathogen that enhance phagocytosis. Upon phagocytosis, the phagosome is fused with a lysosome inside the phagocyte and destroyed intracellularly. Phagocytes can also release toxic chemicals that can destroy nearby foreign microorganisms prior to phagocytosis. Other mechanisms, which are also outlined here, are also involved that can help in effectively disabling the foreign microorganism by phagocytes. Chemical signaling is critical in both nonspecific and specific immune defenses. Table 18.2 lists and states the functions of selected cytokines used by nonspecific and specific immune cells. The list here includes interleukins and cytokines. Please familiarize yourself with these chemical signals that are crucial to immune cell communication. Like in a true warfare, they are the encrypted secret codes of immune cells in launching a highly organized defense strategy of the immune system. Inflammation is the local response to infection or injury. The functions of inflammation are to destroy or inactivate foreign invaders and to set the stage for tissue repair. The key mediators are the cells that function as phagocytes. As noted earlier in the previous slide, the most important phagocytes are neutrophils, macrophages, and dendritic cells. Inflammation is an innate multi-step defense response induced by invading pathogens, but can also be produced by a variety of injuries by heat, chemicals, and physical trauma. Assuming that the infection is local, the summarized steps of inflammation shown here in this slide begins with vasodilation of blood vessels and increased permeability to blood proteins in the area. This is then followed by chemotaxis of white blood cells into the area, which includes the process of margination and diapedesis of leukocytes into the site of infection. Phagocytosis of pathogens and cellular debris then follows that eventually leads to hemostasis and tissue repair, as outlined by the previous slide during phagocytosis. The non-specific innate defense components and mechanisms of the body is not as simple as has been presented in this video lecture. The process of non-specific immune defense is very complex and generally works together with the specific adaptive immune mechanisms. We will attempt to demonstrate these complex mechanisms later with separate videos. Thank you.